welcome to today's webinar, Emerging Trends in Nanotechnology. I am Dr. Himanshu Gupta from Department of Medical Biotechnology, MGM School of Biomedical Sciences, Navi Mumbai. I take this opportunity to acknowledge our Honorable Vice Chancellor, Dr. Shashank Dalvi Sir, Honorable Pro Vice Chancellor Sir, Dr. Nitin Kadam, Registrar, MGM IHS, Dr. Rajesh Goyal, Dean, MGM Medical College, Dr. Narshetty, and Director, MGM School of Biomedical Sciences, Dr. Mansi Thakur. I also welcome our esteemed speaker, Dr. Ravi Shankaran Sir. A warm welcome to all the delegates. I request Honorable Vice Chancellor Sir to say a few words. Over to you, sir.
Thank you, Himanshu Gupta.
Welcome.
On this occasion of webinar, Dr. Ravi Shankaran, Dr. Mansi Thakur, Dr. Himanshu Gupta, all the delegates who have joined for this particular webinar in relation with the nanotechnology and its practical applications. And I am sure with the experience and the knowledge Dr. Ravi Shankaran is having in this particular field, this webinar will be of quite use. I will not come or take much time in your academic venture. All the success for this particular webinar. Thank you very much and good luck. Thank you.
Thank you, sir.
Thank you, sir. Thank you, sir, for your encouragement.
Thank you, sir.
I would now request our director, Dr. Mansi Thakur, to say a few words.
Good afternoon, everyone. First of all, I would like to thank our Honorable Vice Chancellor, Dr. Shashank Dalvi, our Pro Vice Chancellor, Dr. Nitin Kadam Sir, Dr. Registrar, Dr. Rajesh Goyal, and Dean, Dr. Narshetty from MGM Medical College, Navi Mumbai. I would first like to thank our management, Honorable Vice Chancellor, who have always supported us to do all these type of national webinars online at present during this COVID pandemic. Apart from that, I am really thankful and grateful to our speakers who have accepted my invitations, our invitation from Department of Biotechnology. Dr. Ravi Shankar sir is already available, Dr. Priyanka Sabarwal, and Dr. Kamal Prasad sir. So as you all know, the audience who are present available here, you all know that this is the emerging trend in nanotechnology. The nanotechnology is recently, within few years, it will come up as a very great emerging technology, which will be now having a vast application in various fields, like in diagnostic, environment, we have in engineering. Every small things involves the nanotechnology. So I thought it is the right time for our students who are available at present. They are available in online session. So the great speakers, we have an eminent speaker with us who are having a vast experience in nanotechnology. They have designed many, they have transferred the technology to the society, and it is a great opportunity, especially the audience who are the students. You will get an insight here so that you can motivate yourself to go for the nanotechnology in the field of nanotechnology. So I'm not taking much of time. I request Dr. Himanshu Gupta to take the session ahead. And I'm really thankful for the audience as well as the students, my dear students who are available here. And I hope you will enjoy the session, and any doubts, anything you can ask the sir and ma'am, and you will get the opportunity for that also. Thank you and all the best for that.
Thank you, ma'am. I'd like to introduce the first speaker, Dr. Ravi Shankaran Singh, sir. Sir is a professor at National Center for Nanosciences and Nanotechnology, University of Madras, Chennai. Sir is a doctorate from the Department of Analytical Chemistry, University of Madras. Sir has worked at 4 international universities like the Laboratory of Biophysics Sensor Technology, South Korea, Department of Applied Chemistry, Japan, Art Science and Technology Center, Japan, International Center for Micro Nanotechnology, USA. He has visited several foreign countries as part of his research activities and has established the nanobiomaterials and the sensors laboratory as the National Center for Nanoscience and Nanotechnology, Madras. Sir's current research involves development of sustainable and multifunctional nanobiomaterials and nanobiosensors for affordable healthcare applications. Sir's topic is nanobiotechnology for point-of-care diagnostics. Over to you, sir. I would like you to start your presentation, sir.
Thank you. So thank you once again, Dr. Manshi and Dr. Imanshu and the MGM institution and the Vice Chancellor for this great opportunity. In fact, I visited the MGM institution almost 4 years back. I had the opportunity to discuss with Dr. Manshi and the Vice Chancellor also. It's a very good institution, especially on medical. Now today my topic is on nanotechnology for point of care diagnostics. I am from the University of Madras, Professor in Nanoscience and Technology. As we are all familiar with the scoreboard of cricket, this IPL is about to start. Unfortunately, in the recent days, for the last few months, we are familiar with the new kind of scoreboard. The main player is coronavirus. So every day, every moment, we are getting the update on the number of new cases, the number of treated, the number going to the hospital, and going back to the home. So this coronavirus made us to come to a new life, what we call a new normal, where every day our health has to be monitored, where every day we have to take care of our health, where every day we have to take care of our hygiene. I believe that in Mumbai, people like these from the hospitals or the government representative comes to your home, check your body temperature and your oxygen level. In Chennai, we have a regular visitor of municipal person used to come and check our temperature and see whether we are all doing well. This is now part of our Indian lifestyle now. The story here is that why we are very much worried about the coronavirus. When we talk about coronavirus, then naturally the discussion also comes with a nanotechnology. Why is that? We look at this virus. This is the virus, especially for coronavirus, because it has that crown shape. We call it a coronavirus. Normally, virus contains nucleic acids, either DNA, RNA, and proteins. This particular virus, SARS-CoV-2, has a single standard RNA genome and a nucleocapsid of helical symmetry. It's a very simple structure. But the issue is that its size. When you look into its size, it comes almost in the range of a nano. I believe that most of you have some idea about nanotechnology, where we discuss about materials at the nano range. It comes around 1 to 100 nanometer. Since virus comes in the range of nano, so there is always more interest to study how this nanotechnology could help, could explore the options to or opportunity to deal with the virus. Here I am showing the nanoscale. When you see that from 1 to 100 nanometer, you have regular chemicals, molecules, then antibody, virus, and then comes the bacteria. When we take our body, the human body, a normal grown human body like us, we have almost 75 trillions of cells. Our life is made up of two kinds of cells. One is cell phone, that is smartphone. Without smartphone, now life is no meaning. So we have come to that level. The other one is the cells which we are made up of. So when we say 75 trillions of cells, each cell has its own subconstituents. Cytoplasm, DNA, RNA, so many things are inside. So the 75 trillions of cells, we can call it the 75 billions of micro machines. To be healthy, today if you are happy and healthy means that these 75 trillions of micro machines are working. They are living. They are healthy. But when you look into the size, the subconstituent of the cell as I mentioned earlier, it comes in the range of nano. So any issues which we encounter at the cellular level, at subcellular level, if we are able to monitor, if we are able to diagnose, if we are able to treat it, then probably we can take care of our health. Because any disease condition starts from the cells. This is the beauty of nanotechnology, because the size range of 1 to 100 nanometer, which almost comes to the range of the sub-constituents of the cell. So there is much interest to study the interaction of these two, or we can explore the human system with the use of nanomaterial. That is why there is a great interest in nanotechnology, especially to biomedical applications. Now, the screen you are seeing a sugar cube and a sugar powder. If some of you are not familiar with the chemistry of nano, in this I want to give you a simple explanation. You have sugar cube and sugar powder. I believe that most of you have the opportunity or have the habit of preparing tea in your home, or at least your mother or father. So when you prepare tea, you have to mix sugar with hot water. You have two options now. One is sugar cube, the other one is sugar powder. Among the both, if you mix sugar powder, it will dissolve very quickly, faster than the sugar cube. The reason is that compared to the sugar cube, the sugar powder has more surface to volume ratio, surface area to volume ratio. Because even the cube also, even though it's the same sugar particle, but the availability of each sugar particle to the water is minimum compared to the powder form. So this is the main reason when you go from the bulk material to the nano material or smaller material, we tend to increase the surface of the material. However, the volumes remain same. Volume remains same, however, the surface area increases. Any chemical reaction or any biomolecular interaction happening between the these two species, A and B, naturally it depends on the surface area. If there are more surface area possibility, there is more possibilities of interaction. In this case, the interaction is for dissolution. Sugar is to dissolve in water. So the dissolution is now enhanced just because we increase the surface area. So this is nanotechnology. When you move from a bulk material to the nano size, smaller size, there is an increase in surface area that enhances its properties. Whatever the properties, chemical, mechanical, optical, electronic, whatever it is, there is a dramatic change in these properties that we are exploring into the various applications like healthcare. This table shows very clearly, if you have a cube, if we cut into smaller pieces, naturally the surface area increases. So compared to a small cube, and when it will come to the very smaller fine particles, you see that the difference in the surface area. So if anybody asks you the question about what is nanotechnology, just explain smaller size, more surface area, newer properties. Now, of course, today, currently, everybody is worried about the testing, especially with the virus. So all media, all organization talks about testing. And since most of you are biology students, you are very much familiar with this RT-PCR. This is the standard test now being used to test the coronavirus. If you look into the procedure, first you have to collect the sample, and then you have to extract the RNA, then convert into complementary DNA, then amplify, use it with the PCR machine, and you get the results. Because of this multi-stage procedure, it takes at least 1 day to get your result. In the case of coronavirus, if somebody is suspected is having coronavirus, if you allow the person to interact with other people for even 1 day, it's too dangerous. So he may spread the coronavirus to other people. So there is a need that we should test and get the results on the spot. That is where we are interested to do with point of care diagnostics. That means that when everybody craves about fast, the best probability is to do online monitoring, on-site monitoring. Is where the rapid test kit come. Unfortunately, most of you maybe know the story that ICMR, with the advice of ICMR, all the states bought fast kit, rapid test kit from China. After the arrival, when they used it for testing, they found that most of the results are not reliable. Then, of course, they have to stop and then again go back to the PCR testing. So the issue here is that we need a rapid test kit. However, there is a possibility of this kind of false positive and false negative results. All the reliability. So what is the opportunity we have with nanotechnology? That's what we are going to talk today. Because we know that today we need a rapid test kit and how this nanotechnology can help. I believe that we've got some idea of the importance of rapid test kit. Now, before we go to the rapid test kit, first we need to know sensor. What is a sensor? Most of you now, after finishing dinner, lunch, you are sitting there, or some of you may be taking lunch during this session also, because it's comfortable when you're sitting in your home. So when your father or mother, they prepare for your lunch, you get a sweet aroma taste, isn't it? From the aroma, you will understand what is the preparation going on in your kitchen. You know, how is it possible? It's because something you have a special recognition ability that recognize that whatever is happening around you, the smell, the sense, the heat, the cold, you can recognize that. That since we are doing it on a regular basis, we don't realize that it's a very normal process. Whatever is happening around us, we sense that, and then we give some quantification. So there are two major parts involved here. One is recognition, other one is transduction. So when you have a biomolecule, a biorecognition part in the sensor, then we call it a biosensor. Why we have to go for biosensor? Because we know that all the biological processes happening inside of our body, it's all very, very selective. The receptors will attach the particular ligands. Antibody will attach to the particular antigen. So this makes the systems highly specific. So when we develop a sensor integrated with a biorecognition molecule, probably you will get a highly selective sensor. Now, look at the possibilities. We have biomolecules and then transducers. The biomolecules, we have enzymes, antibodies, nucleic acids, and receptors. The transducers, we have electrochemical, piezoelectric, optical, and thermal. So there is a possibility. You can have different kind of combination by which you can make, for example, antibody-based electrochemical sensor, antibody-based optical sensor. Throughout the world, scientists around the world are working on different aspects of this, different combination of this. So naturally, we expect thousands of papers are published every month around the world on various aspects for various applications. Now we will go a little bit deep into the biosensor. The analyte, which you want to detect, for example, glucose in your blood means glucose is an analyte. The blood is the medium, the sample. And if this is going to interact with your detection system, there is the recognition. Maybe a glucose oxidase enzyme. Recognition, your analyte comes and interact with your recognition part. Then the signal will be produced. Based on the signal can be amplified with using a particular transducer, maybe optical or electronic. So this is the basic of any biosensor device. When you look into the bioregulation molecules, we have antibodies, enzymes, cells, or polymer type. You see the structure. Each one has different structure, and each one has its active sites, active binding sites. The issue here is that when you develop a biosensor for your application, you need to immobilize your biomolecules on the sensor surface. When you immobilize your biomolecules on the sensor surface, naturally, you expect that your antibody or enzymes are active on the surface. But unfortunately, I will show this. You look at this. When you have an antibody on the surface, it's not always stand up like this. Sometimes it may be flipped like this. So in this case, you have an issue that even though antibody is on the surface, it may not be available for interaction. So there is a importance that you should understand. We should take care that how we immobilize the biomolecules on the surface, because we have different kinds of biomolecules. We have to adopt different kind of immobilization methods. So we have three types: enzymes, antibodies, receptors. Here, if you take enzyme, it's very familiar to all of you because we know that glucose sensors is commercially available. The substrate we use is glucose, where the enzyme glucose oxidase can be used. This will be interacted with your glucose, and then you will get the product. This is one kind of example of an electrochemical-based glucose sensors. The other one may be a receptor-based sensor. In this case, we take a receptor. Receptor is taking part of our system. It's always specifically attached to the ligand. So, for example, here we have taken the dopamine receptor. This dopamine receptor interacts specifically to the dopamine. So, based on this interaction, we developed a sensor for dopamine. This uses the concept of receptor ligand interaction. The earlier one is, this one is the enzyme ligand interaction. So, as I mentioned that we have a different aspect. The other one is the immunosensor. In this case, we use antibodies. To take antibodies, the interaction of antibody with an antigen gives an immunoreaction. Based on the immunoreaction, we can go for the sensing of the particular analyte. If you look at the structure, I'm going to explain a little bit about this immunosensor, one system. We have three systems, so I'm going to explain one system because of time restriction. Now we see that this antigen binding site is here. So when you immobilize this onto the biosensor, you expect that antigen binding site should be available for binding with your antigen. That's the challenge we have. So based on this, the issues we are facing with the preparation of biosensor, people around the world develop, scientists around the world develop various methods to immobilize. I have categorized in this particular review articles, we have categorized into different formats so that it's easy to be understood. The most used method may be physical adsorption, self-assembly, it's Dextran based or protein A and G, biotin, streptavidin, and then polymer or Sol-Gel or LB method. So what is the desired features? It should be simple, like we expect every time. It should be simple, it should be fast, it should be stable, and of course biocompatible. The most important one is the oriented immobilization so that the analyte is available for interaction. And of course, there should be no non-specific adsorption. So these are the methods available. You can choose whatever the options are depending on your analyte, depending on your application. You can choose whichever is suitable for you. This is what we explained. What is the importance of oriented immobilization? In this case, you believe that you've injected antibody, you believe that all antibodies are on the surface. Of course, it's on the surface. However, how much is available for binding is the issue. So compared to these two, so it is better to have always this optimized oriented surface than whatever the analyte you are injecting will be of better response. There is another problem we face is non-specific signal. We have antibody. We expect that your analyte comes and interacts with the antibody at the antigen binding site. There may be an opportunity, a chance where this your analyte come and interact not at the antibody surface, but maybe on the base surface. Here also, since it is attached on the base surface, if the reaction is based on the mass, probably we get the reading, positive signal, because it is on the surface. However, it's not specific. That means we are getting an error here. The other possibility is that there may be another analyte which have a similar characteristic to your analyte. For example, if you take glucose, if you take an ascorbic acid, folic acid, like this, if you have a similar composition, sometimes that may also come and interact the same site. You are originally looking for ascorbic acid, but something other than it come, it is having a similar structure may come and attach here. That's the problem. So either way, either this case, because it's attached on the surface, not on the antibody. Here it is attached on the surface antibody, but it's not the regular the analyte we are looking for. So either in both the cases we get signal, however, it's not specific. There is an error signal. That is where we are facing the problem with the rapid kits, what we are in the recent imported from China. So when you develop a sensor, these are the features, these are the important characters when we develop. If you are able to make it at this format, where your analyte can come and interact only to your antigen binding site, then probably you will get a very perfect sensor. What we need currently is an assured system. WHO recommend affordable, sensitive, specific, user-friendly, rapid, equipment-free, and delivery to the patients. Any sensor which we develop should be operated by the patient without the need of any technical expert. If we are able to develop that, then that is what not only the WHO, everybody wants that kind of sensors. We name it an assured sensor. Then when you go for the assured type of system, what are the opportunities that nanomaterials are given? Because we know that nanomaterials are very small in size, more surface area. It gives with new physics and chemistry chemical properties that gives more possibility to enhance the sensitivity as well as selectivity of system. Because of its size, it's easy to make it a smaller devices which can be mass produced for various applications. This is one simple example I'm showing. We prepare silver nanomaterials, silver nanoparticles. You see that there are 4 silver nanomaterials. All are silver, but all are in different color. This is a PhD work of my student. This one is spherical rod, then square train. Just because there is a small difference in the shape, you get entirely a different property, even though it's a silver nanoparticle. This is the beauty of nanomaterials. So if you are able to make a small variation, probably you can get a new property. That property can be explored to develop sensors. You see that a simple example where we used this property to detect copper. The process of copper, silver nanoparticle gives a color change, and we found that it's very specific. The simple color change can be visible. So by naked eye, we can see that color change. If the color change is concentration dependent, probably we can make a very good sensor, and we understand the concentration of the analyte also by naked eye. The advantage of this particular system is that we are able to regenerate the surface by simple complexation reaction. It can be used multiple times. Where we are moving from the conical flask to the microfluidic jet. This one you are familiar. In your chemistry in your laboratory, you are mixing with chemicals with this. And slowly we are moving towards the microfluidics. In this case, we don't need to take excess amount of analyte. We take 1 ml of blood, it's more than enough to do multiple analysis. That's the advantage of this. The patient with diabetic condition, he has to check his glucose level at least 4 or 5 times a day. So every time if you take 10 ml of blood from his body, then you can understand his condition. So we need to find a mechanism by which very minimum amount of a sample can be taken. Because in the case of water or urine, there is no issue. We can take as much volume. But in the case of blood, especially in the places like brain, so it's very difficult to get more amount of blood every time. So when you go to the microfluidic type, naturally, you need a very minimum amount of a sample that can be used for your analysis. Simple example, which this work we have done in when I was in Japan. So we developed this kind of microfluidic system. So you can inject your blood or urine, whatever it is. There are 8 channels. If you immobilize your sensor here, you see that, as I mentioned you, the sensor can be immobilized. Now, if you immobilize with different antibodies or enzymes, which correspond to different diseases. So with the flow of 1 drop of blood, since we have different sensors arranged, probably you will get to know your health condition, different kind of health conditions, in 10, almost 8 health conditions at 1 drop of blood. This is the advantage of when you go with the microfluidic system. Simple example shown here, the graph. You can have some control. So when you flow your blood or urine, if you based on interaction, you get reading, multiplexed reading that allow you to analyze with the minimum sample and the maximum number of analysis. We call lab-on-a-chip technologies. This technology has advanced so much. Currently, we are able to develop this microfluidics using papers. The small paper, the papers which you use for regular writing can be developed into this kind of microfluidics using its simple chemistry of hydrophobic and hydrophilic system. The channels can be created that can be used for production. The advantage is, again, because we are just using papers, it's totally going to be low cost. And since it is a paper, you can easily burn it, because disposal of biomaterial is a very important issue. If you take paper, then probably you can just burn it. This is where we are moving towards this paper-based diagnostics. And this is a very simple example, your pregnancy testing. A pregnant woman has some particular protein like SCG. So when you have 1 drop of urine from a pregnant lady, flow it over the surface like this, and then you will get the mark here within a minute. Probably now the pregnancy tests available in the market will show the results in one or 2 minutes. So this kind of sensors we are looking forward. In the case of coronavirus, if we have this kind of system, it's very good. That's what we are looking forward. So this pregnancy testing is the most successful a combination of nanobiotechnology. Based on this only, they are now developing the sensors for coronavirus. So unfortunately, the features which I developed, which I mentioned earlier, the non-specific, the reliability, interaction, this has to be optimized to get that very reliable results. So in our laboratory, we are developing this kind of materials, like the other one is the nano gels or nano papers. It can be immobilized with your nano material, and which shows the visual colorimetric sensing. This for the case of mercury, you can see that color variation. So what next is that, since all of us having a mobile phone, especially smartphone, that means that we all have one instrument in our hand. The smartphone can be used as an equipment. If you develop this kind of point of care system and integrate with your smartphone, probably you have one laboratory in your home. In every home, maybe there are 4 or 5 smartphones available. So that means that we have 5 instruments available in the laboratory. We know that importance of instrumentation. So if you have 5 instruments in your home, then that will be very advantageous. But what we need, develop sensor which can be integrated with, which could be integrated with the smartphones. The best way is to make this kind of calorimetric sensor, which can be easily integrated with your smartphone. Now all the softwares which are being introduced with the smartphone companies and the power of a camera, we can develop huge variety of highly appreciable sensors for this kind of applications. When you apply this,
Oh,
maybe wearable sensors. So when you have the sensors, now if you are able to wear it or attach to your clothes, your body, probably you will get a further advantage where you can be in the home or you can be in the office, you can monitor what is happening in your home, especially when young ladies, young kids, or the old people, if they are left alone in the home, then probably we can monitor with this kind of sensor which are attached to their bedside or into their cloth. The response can be easily with the Wi-Fi systems can be transferred to you or to your neighboring police station or neighboring healthcare center, so that anybody who is near to your house can be taken care, take care of the issue. So again, the issue here is that how to make this. But to make the sensor, what we need is that recognition and translation. That is the very base. So when you go into different formulations, different formats, these two we cannot change. That has to be there. But by playing with the materials, we can try to make it like this. So when you go for the kind of flexible sensor, probably you know, in the form of the ink or tattoo or the films or membranes. So you can have your recognition molecules impregnated with your films or your fibers. Now the issue comes is when you are using a wearable sensor, there is more possibility that the stress and strain, this may be pressure or the environmental factors, that is to be taken care. For that, your material what you are developing, even under the stress and strain condition, should withstand without losing its property. That should be there. So again, similar idea, we have developed a flexible strips like this using nanomaterials, which is impregnated into a polymer. Now it's conductive. Most of the conductive polymer, conductive films are rigid. If you are able to make it flexible, probably now you are don't need to worry about the brittleness. It can be worn. As I mentioned, that we need to do a lot of optimization to bring it to the format of this nano formulation, the form of films, and then make sure that its property remains even under the stress and strain condition. This is another example. You see that this is a nanofiber. This looks like a small kerchief, a piece of paper where we have loaded with curcumin. Curcumin is the main component of turmeric. This curcumin can interact specifically with the lead. So you see that this is the nanofibrous mat. We prepared it by electrospinning method. And then we loaded with curcumin. The color becomes yellow because that's curcumin color. It's a nanofiber mat. Then when we immobilize, when we interact with metal ions, we see that different metal ions, only with the lead, it has a good color change. That shows that the curcumin loaded nanofiber mat interacts specifically with the lead. So it can be, now we can make it kind of blended. You can wear it or fix into the body, or you can just put it over your skin to understand if there is a blood in your your sweat contains the blood, lead. Or can be used as a breath analyzer. If you breathe out, keeping this like an handkerchief, if your body contains any diseases, depending on the disease, there may be some active components volatile organic compounds comes out of your breath. So that can be monitored. This is another method, the same, we are making a kind of a film that shows the color change even with the pH variation. It's a very well-known chemistry that the form is different. So we can make it a kind of a food package system. Due to the decay of food, if there is a change in the pH, naturally you will understand that the food start decaying, that the color change can easily give you an idea that how much the decaying is taking place. To go to the other form, instead of naturally when we take about wearable sensor, that means that polymers or the cotton. So we take cotton means it's made up of cellulose. Instead of cellulose, we can go to cellulose nanocrystals. If you go cellulose into cellulose nano form, again we know that the nanomaterial, because of its nano property, it will get wonderful properties that could be applied for various applications. In case of drug delivery, diagnostics, the cellulose nanocrystals are widely used because it's biocompatible. So you can make any sensor, a diagnostic system, a drug delivery systems which can be can be taken inside the body. So we have taken this kind of system from the cotton waste. So we prepare cellulose nanocrystals again to make this kind of film, which could be further functionalized with your active components that can be used for sensor applications, which can be wearable. So what opportunities we have, which can be useful for continuous monitoring, online or offline monitoring. So we can assess the performance of the soldiers or the sports persons or old person in the home. It gives the real-time information. Of course, it can lead to a home-based personal management. What we have to worry about is its toxicity. And as I mentioned, the stability of the material under the different stress and strain conditions. The other application what we are discussing about the wearable sensor is now the whole country is wearing the mask. Mask, we are wearing it to avoid the spread of coronavirus. There are two ways. One is that we are stopping it, coronavirus from outside, and one from your side. So if there is any material which could respond to the coronavirus, if a sensor, then probably either way you can check. Even if you have coronavirus, you can understand from your mask. Or if somebody next to you is having coronavirus, that can be also understood from the outside of your mask. So all you need to do is to functionalize your mask, make it a functionalized mask, which can detect the presence of toxic compounds. There are reports for using this into various applications, especially for pesticides and other metal ions. Now, since we are under the compulsion to use mask, if there is any possibilities that we can make it a functional mask, you can understand the coronavirus by yourself without need of any diagnostic system. But of course, it needs a little more time to come to the market, but it's a very wide, highly opportunistic area. Anybody interested can move forward because it's easy to make also. More than it will be antibacterial or antiviral and should be sensitive. The similar concept, we have developed some kind of nanofibrous mat. You see that this mat shows specific color change when it's interaction with aldehydes. When we have a cancer in our body, so some kind of aldehydes will come out. We know that our biological system, whatever we are made up of food what we are taking. So depending on each person, the health condition, we expect that some kind of gases will come out of our body. So when the breath analysis, we always familiar with the ethanol sensor with the policeman used to have. And we always know carbon dioxide may come. But along with that, hundreds of gases comes out of your breath every time when you breathe out. If you have a very specific sensor, which can recognize the the volatile organic compounds present in your breath, probably that will be a very good monitoring system to understand your health condition. Of course, it's non-invasive, so it should be highly advantageous for that. Here also we made something like this. We found that able to detect aldehydes, the breath analyzer which can detect the presence of aldehydes in our body. And to make this, we have another systems where ionic liquids. These are very famous using ionic liquids when you talk about the green chemistry or green technologies. This very stable liquid, it's a very low vapor pressure. That's why it's maybe used as a green liquid. So we have used this green liquid, green reagent. We immobilize the biomolecules or our antimicrobial agent. It has been tried to develop a textile fabric that could be used for antimicrobial fabrics for soldiers. It's a part of work I did when I was in US. So for the US Army, our laboratory has developed something like this. We know that the soldiers, they have to have their clothes with them. Probably they may not, sometimes they may not have time to change the clothes regularly. So the antibacterial cloth is very essential for them. So this is the advantage. The same can be applied when you go for the recent demand of masks. The other diagnostic system is imaging, bioimaging. When we go with nanomaterials, normally we use dyes to go imaging of cells. Now with having nanomaterials, most of them are highly fluorescent material. For example, carbon quantum dots. This carbon quantum dots we prepared from solid waste, sugarcane bagasse. This is highly fluorescent in nature. This fluorescent nature of carbon quantum dots can be utilized in bioimaging applications. And because it's a nanomaterial, so bleaching is not there. So it's highly advantage to make imaging systems. We can make it biocompatible also. This is some work, some agencies which described our work actually, the importance of our work. See how we use carbon quantum dots with the cancer cell lines. What is important here is we talk about nanotechnology, nanomaterials, biotechnology, biomaterials. So there is an interface of nanoparticle and biomaterials. Since most of your bio background, you will understand the complexity associated with the biomaterials, dealing with biomaterials. So there is an interaction if we use these two. So this nano bio interface has to be studied very much before we come up with any product for diagnostics as well as a treatment. So there are various factors which you have to do that. The surface area, the porosity, the steric interaction, the receptor ligand interactions, the stability, aggregation, dispersion, various factors we have to understand are optimized when you develop new nanobiomaterials. In this direction, we are showing few things what we are doing in our laboratory because time is almost getting over. This again a kind of flexible electronics where we develop nanomaterials for conductive applications. It's nano-printed resistors which we normally use in our high level sensing. And a kind of contact lens which is loaded with our drugs. This will release the drug in a sustainable way. And of course, we can prepare magnetic nanoparticles. And then this films, which are, you can see this image. This film grows with time and there is change in color. This is highly suitable for food packaging applications. And there is a part of work which are working on with VRC, where we are trying to develop invasive blood pressure sensor. And currently we are working, I'm almost completed with 2 projects. One is from Department of Science and Technology on the development of biochips. Science and Technology, and the other one is Department of Biotechnology on the detection of drug delivery to prostate cancer. I believe that I gave you an overall idea about nano diagnostics, how these nanomaterials could be explored, the properties of nanomaterials could be explored, and the properties of nano and the bio could be combined and integrated to make nanobio materials and devices which could be useful for diagnosis and drug delivery applications. And it has very wide feature for all areas of diagnosis and drug delivery. So with this, I'm completing my talk. Anybody who need further clarification, please feel free to write to me, or my phone number is given here. You can note down my email ID. If you need any clarifications or further information, please feel free to contact me. Thank you very much.
Thank you, sir, for such an educated talk. We now have some questions from the participants. Amla, please go ahead.
Okay, ma'am. There are a few questions asked by the participants. First, do you have a device that can measure alcohol concentration in a chamber?
There is normally we have this, as I told you during this wearable sensors, this policeman who is used to have alcohol sensor. It is available on the market, available on the road. We are seeing that every time policemen are using to check whether you are drunken or not.
Okay, sir. Sir, there are masks that are made from nanofibers as well. Can we please know more about them?
Yes, mask means it's a cloth. That means it's made up of fibers. I showed you the work which we have here, fiber. Here, if you make it into the file, because we have taken in the AFM, it looks like noodles, almost like fiber. So when you make it into the continuous fiber fabrication, it becomes a cloth. That's how our cloths are being made. Now what we have done here, you can see that this is the ultra spinning machine. We made fibers out of the polymers. And now since we have taken it at the FM, we are looking like the fibers, individual fibers we can see here. But we can see in the kind of a mat. If it's prepared continuously, it will be kind of a mat. You can prepare whatever the fiber, any polymer can be used here. Now, yes, we know that we need recognition means we can immobilize. We can enter any kind of active components or chemicals or biomolecules into this, so that it becomes the functional mask.
Okay. So, what are the hazards of nanoparticles?
What are the
Hazards of nanoparticles.
hazards? Because we know that, as I mentioned you, nanoparticles are highly active. Compared to bulk materials, nanoparticles are highly active, highly reactive. That means that we are taking about its active property for various applications. So when you talk about the material, when you talk about the toxicity, it is equally toxic because the toxicity level also will be increased compared to bulk material. Then if you take nanomaterial, the toxicity may also increase because it's now more active now, more energetic. That's why when we talk about nanomaterials, we always worry about the toxicity, especially when we develop materials which we take inside of our body, in vivo sensors or wearable sensors. So that work is now, people are working on that. In many cases, the toxicity is eliminated or taken care by coating with biocompatible polymers. For example, if you take drug delivery system, we take nanomaterials, nano carriers, but these nano carriers are loaded or functionalized with biocompatible polymers. For example, PEG or cellulose. Since these are biocompatible, this will keep this nanomaterial compact and do its function. However, the toxicity of nanomaterials to the biological system, especially when you take the inside of the body, you still need a lot of work.
Thank you, sir. I'm sure there must be a lot of questions more, but due to the shortage of time, we can take them up later or can be mailed to sir directly. We have our next speaker now, Dr. Priyanka Sabrawal.
Thank you, sir.
Thank you, sir. Dr. Priyanka Sabrawal. Madam is an assistant professor at National Center for Nanoscience and Nanotechnology, Kalina Campus, Mumbai University. She's an PhD in nanobiotechnology from Institute of Microbial Technology, Chandigarh. She's an experienced R&D innovator professional since the last 15 years. Madam is working in the area of nanobiotechnology, healthcare, environmental, and clinical diagnostics. She has more than 33 publications to her credit from highly indexed journals, has one patent published and one patent filed to her credit, one technology transfer, and various awards and honors to her credit. She's part of various funded projects. Ma'am is going to talk on nanosciences and nanotechnology in combating COVID-19, market insights, and challenges. Over to you, ma'am.
Yeah, thanks for the lovely introduction. Let me share my screen. Yeah, so to begin with, I would like to convey my sincere thanks to Dr. Mansi for giving me this opportunity to share my views on the topic. And I'll be talking about the various nanotechnology and nanoscience aspects which can be used in combating COVID-19, their market insights and the challenges. As we know, COVID-19, it seems to reach a new tragic milestone every day. And timely diagnosis is the only efficient way to monitor the spread of the virus. And this pandemic has opened avenues to novel technologies which can detect the disease at a much early stage without giving false positive results. And also, it emphasize our preparedness for future to combat pandemic like COVID-19. So I'll be talking about the technologies which are already developed or they are in developmental stage or they have already hit the market. Um, so just to give you a brief about my startup activities, so Biosensica Lab is a recent startup launched in 2019, and our main aim is to develop and market innovative nanotechnology based solutions, which include biochemical reagents, receptor, sensor design, and kit development. And we have a versatile team, me, Kishore Kumar, Professor Mansuri, and Mr. Dinesh Toluja. And our products, they are actually relying on a proven track record of more than 12 years. Only a few representative papers I have mentioned over here, but you can see the consistency in the research work which the team is being carrying out for the last 15 years, 2020, 19, 18, 17, 16, 15, then 14, and 2008 onwards. So the Biosensica lab activities are mainly in the area of bioreceptors, nanomaterial design and development, sensor design and assay development, and also we are into consultancy and contract research activities. So these are some of the listed bioreceptors which we have already developed. We have targeted cardiac markers, diabetic markers, pathogenic bacteria, pesticides, explosive. We have worked on high government top priorities project, prostate cancer. Then variety of nanomaterials have been synthesized and characterized by our team. And we are mainly into optical, electrochemical, and microfluidic, and of course, fluorescence-based sensing platforms. Our clients and collaborators are from industry, academia, corporate side. They are from all domains. And some of the good names are Vital Sense, Genovo Biolabs, Labcare Diagnostics, Affimedics from US, IT Delhi, IT Roper, JNU, and University of Mumbai, the place where I work. I also work for University of Mumbai. So this is one of the, now the previous speaker has already set the platform for the biosensing. I'm not into the going into the details of what is a basic biosensor is. So I'll quickly jump to the diagnostic platforms. So this is one of the interesting award-winning technology we actually developed. It was long back in 2008, but it was for the pesticide detection, and we got a lot of media attention. So as you can see, this pesticide is just a plastic based sensor where the yellow portion is the gold nano coating, and we did laser ablation. So literally we contacted that time the vendor from the Bombay who was doing Ganpati Bappa coating, and he did this coating for us. And that technology was this single sensor was just 5 rupees costing us. And this technology was widely appreciated across India as well as outside India, and we got MIT TR35 award and many Google research and many others. So basic the potential benefits of this technology was first thing is the cost. As I mentioned, it was just 5 rupees cost. It was highly rapid, field applicable, sensitive, accurate, and reproducible. And this biochip was actually based on the electrochemical platform. And from the environmental diagnostic, that was the time when we moved towards the clinical diagnostic area, and we have developed then variety of sensors based on this technology for cardiac markers, for cancer markers. Just to give you a glimpse, because keeping in view today's topic, I won't be able to cover all the diagnostic platforms which we have developed, but this is just a glimpse to give you an idea of what kind of diagnostic platforms we have developed. So we have developed paper-based, microfluidic-based, mobile phone-based, silicon-based, microfluidic-based, column-based platforms. They were developed. Majority of them they have been developed for clinical diagnostics. But yes, we have also worked on bacterial pathogenic bacteria as well. So couple of platforms we developed for the pathogenic bacteria, especially for typhoid. We have extensively worked on E. coli and Salmonella typhi and typhimurium. Then coming to the topic of my today's talk, because we know Corona is everywhere and we all are facing lockdown hurdles, I would say. So let's understand the typical structure of this coronavirus, because if you want to develop any sensing platform, it is must to understand the target analyte which you want to detect on your sensor. So this is a typical structure of the virus, and it has important antigenic proteins, nucleocapsid protein, spike protein, and envelope protein, and also the glycomembrane protein. And majorly the diagnostic people, they are targeting nucleocapsid protein and spike protein because they are antigenic, and they do have I would say earlier also there were some SARS type of viral attacks happened, pandemics happened. So these nucleocapsid and spike proteins of SARS-CoV-2 are very much different, and people have identified the specific domains which are causing such a difference. So mainly the SARS-CoV virus it belongs to a family Coronavidae. The size is 120 nanometers, and the spike, the name corona comes from the spike structure, as it is having the spike structure. And the history of SARS-CoV kind of virus, it dates back to 2003. So this is the virus COVID-19 which we are seeing its impact today. In 2003, we were having SARS-CoV-2, and in around 2012, there was a MERS-CoV. So all these coronaviruses, they have a common ancestral region, but due to the evolution or survival to best theory, these viruses are mutated, or they are man-made, or the mutation happened. It is debatable. So I'm not going into that concern. So, but yes, people are supporting this idea that these SARS-CoV, MERS, and SARS-CoV-2, they have common ancestors that is bad. And in the evolution time, pangolin, camel, or other animals, they were being, they can be the potential hosts. So this is a typical property of any virus, particularly the virus which is having pathogenicity. So it always try to find out different hosts. So, and still scientists are trying to find out from which host these viruses has actually evolved if they are not man-made. And as I was mentioning about the size of this virus, so the virus, it is less, it's around 100 nanometer, which is 100 times smaller than a bacteria, because a bacteria is almost 1 micron, 2 micron in size. So if we want to see the virus, we really need the specialized electrochemical, not electrochemicals, my bad, TM or SEM we need. We cannot see virus with the naked eye or the optical, under normal optical microscope. Then comes the transmission of the coronavirus. They are zoonotic in nature. That means they are transmitted between animals and people. Their hosts are changing. And we all are aware about the various symptoms, or what are the precautions which we need to take during the transmission. I'm not going into the details of the transmission side. But here the important point I would like to emphasize here is, as I was talking about the structure of the virus, so it has S protein, N protein, M protein, and E protein. But if you see the structural similarity of SARS-CoV-2 with the earlier reported coronaviruses, so S protein shows 76% similarity with SARS-CoV, and with MERS it is just 29%, whereas N proteins show a more similarity, and M and E are more closer to other coronaviruses. That is the reason why, if we are working on any diagnostic platform, people want to target N protein and S protein. S protein is better, but the only challenge with the S protein is it's very expensive. 1 mg is initially, I'm talking about 2 months back price, so it was 1 mg was for 1 lakh rupees. So now most of the companies are stepping in. Even in India, raw material is available. Definitely prices, one can get S protein at a better price. Then comes this ACE2 enzyme. So it's very antagonistic enzyme, converting enzyme 2. So this enzyme is actually present in the pulmonary, our pulmonary alveolus. So main role of this ACE2 receptor is that the SARS-CoV-2 virus, it goes and binds with the ACE2 receptor, and direct models people have proposed, where they are suggesting the internalization, how the internalization is happening inside the body. So you can see here the SARS-CoV-2 virus, it has spike protein. So spike protein has the receptors, mainly the receptor binding domain, which actually goes and bind with the ACE2 receptor, which is present in our lung samples, in our lung cells. Then comes the vaccine development. Everyone is talking about when the vaccine will come, when that miracle will happen. And in fact, I was reading one report yesterday that Oxford vaccine is, people have put that vaccine on hold for some reason. The reason was not mentioned over there. But for a better understanding, this is the structure of the coronavirus. And you can see here there is a receptor binding domain present in the spike protein. So spike protein is the whole thing, and in that people have identified only the receptor binding domain, which goes and bind with the ACE2 receptor. So now the people are trying to target a receptor binding protein both in therapeutics as well as in diagnostics. And in vaccine development, we have a variety of options available, like RNA vaccines, DNA based recombinant vector, inactivated and live attenuated vaccines, and every vaccine has different clinical stage. So I was reading one article which says if you start from the scratch for any vaccine development, it can take up to 16 years. But unfortunately, we don't have time for 16 years. So what people are now trying to do is they are keeping the backbone of the drug or the vaccine same, and they are just trying to do the side modifications which are effective enough so that a quick solution can be brought into the market. So once the vaccination it is developed, it passes all the stages, then another question is how to scale up that drug or the vaccine. So these are the different pros and cons of different vaccines which people are using on RNA vaccines, DNA vaccines, recombinant vaccines, viral, live attenuated and inactivated. So this tabulation is actually done on the basis of previously reported coronaviruses. So this coronavirus is totally new. So we cannot rely on this tabular form that which vaccine will be a better format. So only thing we are doing is right now optimized hit and trial we are following. And soon we are hopeful that we will be having a vaccine soon in our hands. Then there was an interesting study done by IIT Delhi based startup, and they are saying that ashwagandha has good property, has good people even I have seen people are taking ashwagandha tablets also. So what is the mechanism how it can control coronavirus? So this ashwagandha, it has a special chemical vithanol. So vithanol and propolis has the cape chemical, and they have the capability to target mpro. So this mpro is actually the viral protein, and this protein is required for the viral replication. So this protein expression is actually hindered in presence of ashwagandha or propolis, and that's why people are taking ashwagandha and propolis vaccines. And one this startup based out of IIT Delhi is taking forward this research. Then a lot of curiosity I have seen in the audience about nano coating for protective personal protective equipment. So I have a couple of slides to show you. So the first technology I would like to discuss with you, it was developed by Jyoti Institute of Technology based out of Bangalore, where they have used the colloidal particles, and they were these colloidal nanoparticles, they are actually organic inorganic hybrid nano coatings, and they are based on silica network. So these colloidal particles, they are actually entrapped inside the silica nanoparticles, which are further coupled with the polymer matrix, and it is extremely hydrophobic in nature, and whenever a virus comes in contact with this extremely hydrophobic nature, it is killed. So specifically, they are doing proof. They are showing proof of concept for COVID-19, and still research is going on. So there is another work done by IIT Roper. They have developed hydrophilic and hydrophobic nano coatings, and they have used actually copper-based nanostructures. And because copper has good antimicrobial properties, and you can see the SEM micrographs where the copper mesh kind of structure is shown. Not details they have given, probably they are filing for the patent, but they are claiming that the surface is both hydrophilic and hydrophobic. Hydrophobic can be used for antibacterial properties, whereas the hydrophilic, they are trying to use for anti-fogging agents. Then another technology for the PPE, it was developed by IIT Kanpur, where they have developed an antimicrobial coating, and they are using the virucidal drugs. So these virucidal drugs coatings, they are using actually polymers which can resist the attachment of bacteria and virus onto the mask, onto the personal protective equipments. Any additional protection will be included to the polymer using these molecules that can either destabilize or neutralize coronavirus or other viruses like influenza. And the combination of antimicrobial polymer coating along with the virucidal drugs is expected to provide a synergistic antiviral effect. Then in another technology which is developed by Nanova Hygiene, and the studies are going on in collaboration with Reliance Industry. So what they have done is they are using silver nanoparticles, as they have been reported to inhibit the replication of the virus nucleotides in presence of silver nanoparticles. And the main mechanism of its being, it's virulent. So the silver nanoparticle, it binds to the electron donor groups of the virus, such as sulfur, oxygen, and nitrogen, commonly found in the enzymes within the virus. And this causes the enzymes to be denatured, and thus it can effectively incapacitate the energy source of the cell. And it can also inactivate the virus by interacting its surface protein molecules. And this technology has very well worked with in case of HIV, hepatitis viruses. Then this is one of the technology actually we have proposed, and we are doing the work actively with the IIT Delhi. And this project we have submitted with the with SUB, and it is under active consideration. So here trying to develop nano coating which will have antiviral properties. So the coatings different. This we have actually designed for the 3-layered mask, and each layer of the mask will be having a specialized nano coating. I'm not going into the details because it will be a separate topic of the lecture. So I just want to give you an overall idea today how the nanotechnology is playing a role in SARS-CoV. So also people have studied about the stability of the virus, and different surfaces like copper, cardboard, stainless steel, plastic. They have been studied for the half-life of this virus. And I understand most of us are following this protocol. If something, even the vegetables or the courier or any paper is coming from outside, we are keeping that material for some time, because by any chance, if a virus, it has a virus contact, so virus should be inactivated. But if you see the half-life of the virus, it has different half-lives on different substrates. Like on copper, it has 3.4 hours half-life, whereas on plastic, surprisingly, it has more half-life. And these important parameters, these are the parameters which are very important to be considered when we are developing any sensing platform, because the surface of the sensing platform we can tune accordingly, whether it is hydrophobic and hydrophilic. And from the reports, it is suggested that the hydrophobic surface has a tendency of showing more half-lives for the coronavirus. Now, few technologies I have discussed with you, but what nanotechnology and nano materials they promise, they are low cost. They have scalable detection methods for the detection of virus particles. They have improved virus disabling air filtration, as like in personal protective equipments. They have enhanced personal protective equipments, including face masks. And nanotechnology is also finding its way in drug and vaccine development, and new therapeutic solutions people are now working on. So these are the different strategies against COVID-19 with medicinal and nanochemistry. You can work on inorganic nanoparticles, polymeric nanoparticles, peptides, self-assembly proteins. And we at Biosensica, we are trying to explore variously modified nanoparticles which can have applications to inactivate the COVID-19 virus, and also certain inorganic polymer-based nanocarriers like dendrimers, micelles, vesicles, carbon nanotubes, gold nanoparticles, iron nanoparticles, mesoporous silica nanoparticles, and even quantum dots. They can be used to develop different diagnostic platforms for COVID-19. And the nanofilms, they are playing a very important role in this COVID-19, because these nano coatings, they can be coated onto the surfaces where this virus can come in contact. And I was reading some articles where in abroad, some companies, they have started making certain paints or the doorknobs. They are coming with some special kind of nano coating which is having anti-viral or antibacterial properties. And that paint, that nano coating is also having application in hospital scenario, where a lot of where there is a fear of contamination, especially inside the OTs. And there was a report from a Chicago hospital that they are going to monitor for coronavirus, especially inside their OTs, for years now. So this emphasizes the need of the development of such kind of nanotechnology based solutions. So so these are the different nanomaterials which we have synthesized and we have characterized, as well as we have worked on variety of metallic nanostructures: gold, iron, copper, zinc, palladium, silica encapsulated, and you can see the TM micrograph. Then also we have worked on layered chalcogenides like black phosphorus, MOS2, WS2. Then carbon-based nanomaterials, graphene, graphene oxide, graphene nanocomposites. Other nanocomposites also we have worked on, like boron carbon nitride, carbon nickel rods, indium based nanostructures for volatile organic sensing. And also we have developed conductive inks which are based out of graphene and CNT. Now there is a question: if we have worked on so many variety of nanomaterials, which material we should be using for which application? So that's a very interesting question. So for optical, suppose you want to explore optical based properties of any nanostructure, say so you want to develop some visual sensor like a pregnancy testing kit, then metallic nanostructures, they are good alternatives. And if you want to work on electrochemical sensor, then you need a good semiconducting material, then layered chalcogenides, they can be good options. So this is the way one can balance out which nanomaterial will be useful in what kind of application. So now, to give you an insight about the global COVID-19 diagnostic market share, so you can see here a majority of the market pie lies with the laboratories which are involved in the COVID-19 diagnostic, and it is followed by hospitals, diagnostic centers, and a comparatively smaller pie goes to research institute. Uh, then comes the COVID-19 testing scenario. So majorly, most of the testing which is being done today is based on either on molecular testing or antibody testing. So molecular testing, a patient's swab sample is taken and then it is mixed immediately with the buffer, which is extracting RNA, and that RNA is converted into DNA. But this conversion, the PCR, which is required to amplify the DNA to actually see whether the person is having COVID or not, it needs a sophisticated instrument or a lab. So samples are collected in the hospitals or the place where the patients or the suspected persons are, and then they are taken to the specialized labs. So the molecular testing, they have a lot of good specificity, but this is the issue, they need a specialized lab. Whereas serological testing, where we are checking the antibody response inside the patient whenever a patient is exposed to COVID-19. So what happens is, here I have seen a patient sample is taken from the needle here, but even a finger prick sample is also sufficient enough, and that is actually taken on a lateral flow assay, on a lateral flow dipstick kit, which is just like a pregnancy testing kit, and our previous speaker was also mentioning about it. So this is a lateral flow assay where we have on the test line, we have control line and test line. So test line is having specific antibodies towards a nucleocapsid protein or a spike protein, and if the antibodies are raised inside the patient body, so they are actually the antibodies, they go and bind on the test line and giving you a signal whether the test is valid or not. And we have developed recently an antibody serological test with our collaborator, and that test is approved by ICMR as well. But yeah, so one thing I would like to add here, because if you look at the graph here, so this graph is very important. On the x-axis you have the date time, days number, I mean the day 0 day when a person gets the infection, COVID-19 infection, and followed by the subsequent days, and y-axis is the antibody level. So this is the 0. This is actually the first day when a person is exposed to the virus and a person gets a fever. So IgM levels are very high, and then followed by the 7th day, IgM and IgG, they are very high, and then they tend to subsidize. So this is a major setback of the serological testing, that if you do the testing before 7 days time, you may diagnose the person that it is not having it is not positive, whereas the person is actually positive. So this is not the limitation of the kits, I would say this is a limitation of the test, because the person's immunological response is not so aggravated or not activated till the 7th day of the post-infection time comes. So that's why now our government is suggesting to test to go for antigen testing as compared to the antibody testing. And antigen testing, people are taking from the swabs, and same this lateral flow testing platform is being used for the antigen testing as well. Then again, the market insight of COVID-19. So just if you look at the CAGR, which is cumulative aggregated growth rate. So this is very important in terms of the investors or the people who are sitting in the market, whether one should invest in this technology or not, or what will be the revenue. So today the CAGR is 17.3, which is very high. I think highest for any diagnostic platform reported so far. And in 2020, we are expecting $3.3 billion of revenue companies are going to make who are working in COVID-19 diagnostic. And this will be going to increase from 3.3 to 8.5 billions by the year 2026 or 2027. And if you see the swab testing, it is actually taking the major pie, 16.5%, and followed by the hospital segment will make 1.5 billion revenue, and the oropharyngeal swab segment will be making 920 million revenue. So, and people are suspecting that Europe will be making more money in case of COVID diagnostics, because they are doing large number of testing, and they are trying to capture 50%, 57% of the market, because most of the diagnostic companies, they are based out of Europe. That may be the reason that Europe is planning to take that flag of capturing highest market pie in COVID-19 diagnostics. Then these are certain kits which are being approved by Government of India. So this is my lab kit, which is the first kit. We all know it was in the media. And then this is the ICMR approved IIT Delhi kit, where it is again PCR based kit, but the price is very cost effective, and they are claiming that they can do the PCR testing in a very reasonable time. It is not taking 2.5 hours time, whereas my lab kit is taking a longer time. And then there is another kit, TruePep, which was initially designed for the Mycobacterium tuberculosis detection, but the company has done certain modification in their technology, and now the same kit can be deployed for the COVID-19 diagnosis. Now, what are the testing challenges in COVID-19? If you see this graph carefully, so again, this is the start of the symptom at the 0 day, and you see the virus in the ear of a patient. It is maximum at the time of the infection, but slowly it goes down. And there is a period here, if you see here, 6 to 9 days time is the period when a person is having maximum when a person is showing maximum symptoms, person is having maximum discomfort, but the virus in the airways capacity is negligible. It's minimized. I would not say negligible, it is minimized. And also the antibodies in the blood, they have not reached up to the peak level. So this is the major testing challenge we have, and these challenges are imposing their impact both on nucleic acid test, our molecular based diagnosis, as well as on antigen body based tests. So these are some of the comparisons of the COVID-19 testing. So the rapid flow, ELISA, and the diagnostics. So quickly to summarize, I would say rapid technologies are very good, as you can get results on site in 5 to 10 minutes. But yes, they do have false negative results. If the test is not done at a proper window time, then a person can give negative signals, a person can be misdiagnosed. ELISA is very robust. You can do multiple samples, you can analyze multiple samples in one go, but the only problem is you need to have 96 samples in one go, and it will take 4 to 5 hours time minimum, because you do have blocking steps or many steps. And then diagnostic RT-PCR, it is all in all, people say, but it also has impacts. It can also give false negative results, as I was showing you that graph. But it is very authentic. The chances of getting false negative is minimum as compared to serological antibody based testing in COVID scenario. Then this is one oh my bad. So this is one of the our technology, which is actually approved by BIRAC, Government of India. So we have proposed a electrochemical based dual antigen and serological testing, where we want to design a chip like this, which will be like a glucometer, and which will be inserted inside a small electrochemical readout, and you can diagnose a person quantitatively. So this will be very sensitive, and idea is if we can do the early detection, because by achieving a very high sensitivity, then a proper clinical interventions can be made at a reasonable enough time, and we can really avoid the spreading of this disease. Then Also, we have designed aptamers. Aptamers are nothing but short oligonucleotides. They work like antibodies, but they are new kind of bioreceptors, and they are actually screened by Select process. And we have recently filed Indian patent for the aptamers for COVID-19 diagnostics. So this is one report we have also published in ChemCom, and aptamer generation is the strength of Biosensica lab, I would say. And we do the aptamer synthesis in a microtiter well. I'm not going into the details of this technology, but you have a reference, and you can go through this reference. Otherwise, you can feel free to contact me for more discussions about the aptamer generation. And also, we have identified the antigenic peptides from nucleocapsid protein and the spike protein. So what are these antigenic peptides? Because the whole protein is very big, and we have screened actually small peptides which are antigenic. So rather than targeting the whole protein, we are targeting antigenic peptides. And then we have done structural structural prediction studies also, and also the physical parameters of the peptides are also calculated. I'm not going into the details of this technology because it is still in developmental stage, but the idea here is that rather than targeting the whole protein, we are targeting the antigenic peptides in order to have more specificity. So once the peptide is ready, then one can develop the bioreceptors, either antibodies or aptamers, I mean, these bioreceptors, they can be used in developing the biosensing platform. Then I would like to quickly discuss with you some of the technologies developed by people across the globe. So this is a selective naked eye detection. This paper was appeared in ACS Nano, where the sample was collected from the nose, and then viral DNA was extracted, and then it was poured into the solution of ASO. Is a gene which can detect the viral RNA. ASO was tagged with the gold nanoparticles, and the gold nanoparticles, if there is a viral gene, the two gold nanoparticles, they tend to aggregate and their color changes from pink to violet. So this essay is actually based on the aggregation studies, and this is very quick. They are claiming that one can diagnose the COVID-19 cases within 10 minutes time, even less than that. Then this is another technology where people are using laser of specific wavelengths. So idea here is, we all know about Raman spectroscopy. So all biomolecules, they give specific signals in the Raman spectroscopy, and you can see here, this is the coronavirus. When you light the coronavirus when you shine the coronavirus with specific wavelength of the light, you get some signals like this. And to enhance the signal, the authors, they have used the carbon nanotubes, and as you can see here, these carbon nanotubes, they act like as a brush structure, and the coronavirus is entrapped inside the brush structure. And the authors, they have successfully shown they can differentiate influenza, parainfluenza, and rhinovirus. And now they are working on the COVID-19 diagnostics. In this technology, this is based on thermoplasmonic sensing. So thermoplasmonic sensing means you have a microfluidic channel, and the gold nanoparticles, they have a very unique property. If you pass certain laser light onto the gold nanoparticles, especially in the IR range, the gold nanoparticles, they tend to heat up. So what they are doing is, they have tagged gold nanoparticles with certain receptors like DNA aptamers, and these DNA aptamers, which are tagged with gold nanoparticles, they can interact with the virus, and once the virus is entrapped, then through the laser light, it can be killed. So now studies are going on if this thermoplasmonic sensing can be used in the clinical treatment as well. So still studies are going on, and you can see here different picomolar concentrations of the sequences are being used, and the signal is detected. Then this is the cell-based biosensor for spike protein antigen. Let me tell you a small thing, that all the cells, they have certain potential. And as we know, these SARS-CoV virus, they can go and interact with our alveolar cells where ACE2 receptors are there. So this is a lab-based sensing methodology which people are trying to do. What they have done, they have reared the Vero cells, which can sustain the SARS-CoV virus, SARS-CoV-2 virus. And these membrane engineered cells, they are having certain receptors where which are having the spike antibody, and when the virus will come in contact with this antibody, the potential of the cell is changed. So when the virus is not attached to the cell will be having different potential. After the specific binding with the antibody on top of the cell, there will be a difference potential. So that potential difference people are now trying to correlate for the diagnostic purpose. Then comes the MEMS technology, which is a futuristic technology, and the MEMS is nothing but a small silicon device which is capable of doing oscillations. So the idea here is MEMS is usually a silicon based sensor where a gold coating, a certain pattern is made up of gold, and on gold, you know, gold is very biocompatible, and on gold the specific antibodies which are specific to COVID-19 virus, either to nucleocapsid protein or spike protein, they can be immobilized, and the cantilever will show certain bending, as you can see here. But the moment the virus will come and there will be an interaction which happens onto the cantilever, which is functionalized with the specific antibodies, this sensor will bend, the cantilever will bend, and due to this bending pattern, and this bending is actually concentration dependent, and this bending is actually corresponding to the presence of the virus in the clinical samples. And the company Cepheid is the company who is commercializing this technology, and they have also made a microfluidic chamber array for PCR. We all know a thermocycler usually takes 2.5 hours to do the PCR amplification. So people are now hunting how we can control this reaction time, because 2.5 hours is a longer time and one cannot wait for the diagnosis of the COVID-19. So people have now developed small microfluidic chambers where the reactions volume of the PCR reagent is actually reduced to few microliters. Already PCR we are doing in microliters, but here the PCR reaction will be in picoliters. So if you reduce the reaction volume, then the amplification time is also reduced. Then there is another technology which is in developmental stage. As you can see here, this is a CMOS sensor array. So this array is nothing but it is having a multiple array of cantilevers. They are aligned, and each cantilever is capable of giving signal. So idea here is if you can integrate the signal to have more sensitivity, because you know in cantilever there's a lot of noisy signals many times. So to avoid that noisy thing, they have developed an array of the CMOS sensor, and this is the device which can read out the signals. And also it has a provision of Bluetooth where one can transfer the signals to the nearby lab or nearby hospital, because transferring of signals and transferring of information or the data, particularly in case of COVID-19, is very important, when each state, every government is keeping an eye on the number of patients which are coming up on daily basis. Then this is a breath analyzer which has been developed. It is being used to detect COVID-19. So as we know, a person who is carrying the coronavirus, it actually releases certain micro droplets which can have the live virus, which can have the viral RNA or the viral RNA microparticles. So not much information is being revealed by this company, but what we have understood from the technology is it is again a nanostructure based technology where certain structures are being designed which can specifically bind with the viral RNA or the virus or the whole virus. Then comes the hyperspectral imaging to study prognostic value of COVID-19 rash. So this is again a very interesting study which says the patients, in most of the patients who are diagnosed with coronavirus, they tend to develop a variety of rashes on their body. And this rashes are people are calling it as a frostbite. They look like a frostbite or the COVID toasts. And these frostbites or the COVID-19 rashes, they are entirely different from other kind of rashes, and this is actually happening because of ischemic phenomena, due to the which is caused by the lack of blood supply to the digits for a prolonged period of time. So this, in this hyperspectroscopy imaging, as you can see the device, if you scan the rash with device, so this device will tell whether it is a COVID-19 rash or not. So this has a lot of application in diagnosing people who are recovering from COVID-19. I agree, in the initial phase it won't be able to tell it. This technology you cannot use in early diagnosis because a rash will not develop in earlier cases. But yes, if a person is already admitted to the hospital and you want to see what is the recovery pattern, then this technology has a very good potential. Then another technology is nano interferometric biosensor. So this is again a microfluidic device, and inside the microfluidic device there are antibodies which are specific to coronavirus, and a certain signal is actually recorded when there is an interaction between antibody and the coronavirus. So again, this is a label free but laser involved technology, and nano interferometry is where the nanomaterials are used, and the properties of nanomaterials are not material dependent, but it depends on the size of the nanomaterial. For example, doesn't matter you use gold, copper, silver, any metallic component you can use in this technology, but the properties are dependent on the pattern, how these nanoparticles, or they are whether nanoparticles or the rods, or what array they have. So another very promising technology, and the sensitivity is very good. One can achieve picomolar to attomolar range. Then another technology, this is based on vibrational spectroscopy. So this is more likely what we have discussed in case of Raman spectroscopy, but this technology is actually developed by Israel people also. They are also working on the same type, where they are saying the technology is based on saliva based methodology. So they are taking, here I am showing the nasal swab, but in the Israel technology they are actually taking the gargle sample or the saliva sample which is mixed, and then they are applying the artificial intelligence to specifically capture the virus, and they are basically monitoring the absorbance or the transmittance, because every virus or bacteria or any other biomolecule, they tend to show a certain pattern of absorbance or the transmittance. Then quickly, I would like to show you some of the clinical technologies which are being developed by us. So this is a technology which we have developed for a cardiac marker myoglobin, and you can see here there is a sensor, electrochemical sensor, which is functionalized with phosphorine. So we were having a collaboration with University of Manchester. So phosphorine we received from them. It's very difficult to exfoliate the black phosphorus because it catches fire. So then we were the first one to develop a bio interface on the black phosphorus on the phosphorine. And you can see here we did the cost analysis of other myoglobin sensors also. The detection range which we achieve in electrochemical sensor was huge. So this is the main point I would like to emphasize here. If you really want to have a very good sensitivity, then electrochemical sensors are the best one in comparison to optical or the fluorescence spectroscopy. This is a quantum dot based assay which was developed for the cardiac marker myoglobin. This was actually the work of my PhD student, and today he has submitted his thesis as well. So this is again the myoglobin aptamer and the carbon quantum dot, which is functionalized with myoglobin. And we were working on excitation and emission studies, and whenever there was an interaction, the excitation or emission, it is actually shifted. The more details are available in the reference which I've given over here. And this is a surge based aptamer, where we have used surface enhanced Raman spectroscopy. Again, This was done for myoglobin. I have done extensive work on cardiac markers, myoglobin, troponin, BNP, and FBAP. So here we have exfoliated WS2, and the WS2 was actually immobilized on the silicon substrate. And because it has sulfur groups, so gold nanoparticles were tagged by using disulfide linkage. And on top of that, aptamers, which are specific towards myoglobin, which we generated, were coated, and in presence of myoglobin, which is present in the patient sample, this interaction was happening and we were getting specific results. So if you are not working on aptamers, you don't add aptamers, and you add patient sample, the specificity you won't get. So for getting specific results, aptamers we have added. And another important point, in SIRS, you cannot work on antibodies because the antibody molecule size is 16 nanometer, which is creating a steric hindrance in Raman spectroscopy, whereas aptamer is just 2 nanometer or 3 nanometers. So aptamers are giving us an edge or advantage in terms of size as well. This is another platform which was developed by us. So this is for the bacteria. So we are selecting first aptamers in a microtiter plate, and these aptamers were screened against the pathogenic strain of E. coli, and once the aptamers are ready, they are immobilized on a commercially available sensor. So these days we are fabricating our own sensors also, but this one is a commercially available. And in the schematic you can see here, this is a nanomaterial, and the blue one is a linker, red is the aptamer, and this is the whole bacteria. And this is a label free technology of detecting the bacteria, and this can give us very good results and specificity. You can see here, the aptamer which we have generated, it was against E. coli O78, and we were getting maximum signal, whereas for other E. coli strains we were not getting enough signal. So the sensor which was developed based on this nanostructured apta sensor was highly specific. So this was all about from the technical side from my side, and we have got couple of awards and a lot of media coverage in the last few years. Then we have done extensive training. We have trained postdoc fellows, PhD fellows. We have guided postgraduate. Also, we have guided. This list is not updated, so I'm sure the numbers will be more. And also the BTech and undergraduate students. And Biosensica Lab is open to collaborate in having joint PhD students, MSc or project fellows, or we are open to write joint collaborative research projects if the idea is novel, or we can work together with the scientists or the researchers or the clinicians to come up to a innovative idea. We can also work as a consultant. We can solve your problems, and also we are working on the turnkey projects. These are the couple of areas where we are finding our strengths. We are having strength in nanomaterial synthesis, biofunctionalization, bioreceptor, especially aptamer design and synthesis, sensor design and fabrication, biosensor development, bioreagents development, affinity purification, assay design, microbiological studies, bioseparation, microfluidics for finding biological applications, and point of care devices. And since a lot of students are attending as audience today, so our internship applications are also open. And if you want to join us or do internship in our lab, please contact me. And I will stop here. This is all from my side today. And now the forum is open for the discussion.
Thank you, Dr. Priyanka, for such an
Yeah.
informative and an updated talk.
Yeah.
I'm sure there must be a lot of more questions coming in from the audiences.
Yeah.
Amla, you can please proceed.
Okay, ma'am. Ma'am, you mentioned about coatings into the mask. Does wearing such a mask actually affect the breathing ability?
No. Actually, it's a very valid question you have asked. There was a paper which appeared in March or April this year, and they were talking about garlic essential oils. And people were planning, in fact, we were also planning to use garlic essential oils. So there is an issue always. The garlic has a essential oil which has some aroma. So people are always working on those nanomaterials which don't interfere with the breathing capability, and there should not be any aroma coming out of the nanomaterials.
Okay, ma'am. Ma'am, there's one more question. Why breathing analyzers are not successful for COVID-19?
See, because it's a respirable disease and a highly communicable disease. So the issues of disinfecting the device every time after every use is very challenging. And if a person is an active carrier and the person blows the breath into the, that analyzer, the person who is holding the analyzer or the nearby person who is sitting next to the person is at risk. That's why the breathing analyzers, they are not gathering a lot of attention.
Okay, ma'am. Ma'am, what are the characterization tests that are necessary when involved in the production of nanoparticles from biopolymers which are to be further applied in pharmaceuticals?
See, it depends on the nanomaterial, but as a thumb rule, I can answer to your question is size is very important. So you can do DLS, dynamic light scattering, and then charges are very important. What charge? As you said in therapeutic side, so charges are very important. So zeta potential is again a good tool can be used. And these are two thumb rule, and then definitely you would like to see the morphology of the nanostructure. So you have to use SEM or TM. Again, it depends on the size. So if the size is big, then SEM is enough. If the size is very small, or you want to see something, you want to have some internal composition idea as well, then transmission electron microscope can also be used. So these are the basic techniques. But in bio, I have seen people are conjugating the nanomaterials with drug molecules or some other biomolecules like proteins and all. Then people do people do gel electrophoresis also.
Okay, ma'am. Thank you.
Yeah. Himanshu, Dr. Himanshu, your mic is mute. Yeah.
I'm sorry. We can take
Yeah.
one last question from Badare sir. Why
Yes.
breath analyzers are not successful for COVID-19?
Yeah, I actually answered to this question quickly. I can sum up because it's a contagious disease which spreads from aerosols. So a person who is holding the breath analyzer or a person who is doing the test can also be exposed, because if a person is exhaling into that. And also there are specificity issues as well, because in breath markers you cannot put the bioreceptors. You have to rely on the nanomaterials itself. So studies are going on to design specific nanomaterials which can only interact with the coronavirus. So it's a specificity issue as well with breath markers.
Thank you, Dr. Priyanka. Thank you so much for your time also.
Thank you.
Thank you. With this, we have the last speaker of the day, Professor Kamal Prasad. Prasad sir. Sir is a professor. To give you a brief on Professor Kamal Prasad sir, sir is a professor at the University Department of Physics and the director of Computer Center at Bhagalpur University, India. Sir has about 26 years of teaching experience in various organizations like the SLIET Punjab, Central University of Jharkhand, Ranchi, and the Aryabhatta Knowledge University, Patna, which is a technical university. Sir has successfully guided 12 PhD and 2 MTech students and is a member of the editorial board of different journals like Colloids and Surface B, and is also an associate editor of the International Journal of IET Nanodielectrics. Sir has over 150 publications to his credit, including 2 books, dozens of book chapters, and 16 X-ray diffraction reference data in the International Center for Diffraction Data, USA. Sir is an active peer of different internationally recognized journals. He has delivered more than 30 invited lectures in different national and international conferences and workshops. Sir's current research interests include synthesis and characterization of eco-friendly ferroelectric, piezoelectric ceramics, as well as ceramic polymer composites and advanced nanomaterials for different industrial applications. Sir's talk is on potential and promises of nanotechnology. Over to you, sir.
Okay, thank you. Good evening, everyone. So myself, Kamal Prasad, as she told, at the outset, I must thank the organizers of this webinar, especially to Dr. Himanshu and Dr. Mansi for inviting me and providing me the opportunity to interact with you people. And as told, in the audience, we have mostly, not audience, as told, the audience, we have mostly students and young faculty members. So I have prepared my slides accordingly. So please forgive me because I am not a professional who belongs to biotechnology side, but I will try my level best to give you something. I am basically physicist, right? I am sharing my slides. We have three things: information, energy, and materials. These are considered to be the three pillars of development of modern society. And among them, the materials plays indispensable role in development of humanity as a whole. This is so. Before going into the thick and thin of the nanotechnology, as a teacher, I must introduce this particular slide to you because this is for students. So basically, there are four components in material science. Uh, one is processing, another is its structure, materials structure, and the on the structure part, only the property which will be displayed by the material, prepared material, will be depending upon the structure of the material. So, so processing has large impact on the ultimate property of the material. So properties which will be displayed by the material, whether it is nano or macrophage or microphage, all depends upon the processing of the processing technique, and the property ultimately decides its performance. As Professor Priyanka and other speakers Ravi told, and they have very rightly said that nanomaterials, and they have certainly explained many kinds of nanotechnological applications in biological side and medicine and others. So this performance of the material will ultimately depend upon the property. So structure have large effect on the performance of the material, and ultimately it is processing which decides the performance of device or system as a whole. So being students of material science or nanotechnology, you must keep in mind that processing plays very immense role in the performance of system or devices. So I am showing one example that you might have seen three beads here of alumina oxide. One is transparent, another is translucent, and third one is opaque. This is only due to the processing difference. So one process gives you transparent, another gives you the material transparent, same material, alumina oxide, and third one gives you the opaque one. So processing of material plays very important role. Please keep this in mind. And nanotechnology is everywhere. It is available in nature, as we know. So this is very famous lotus effect. You must have seen that droplets dew are not sticking to the surface of the leaf. If you put it in under the microscope, then you observe such type of structure of this leaf. And if you magnify those, then you will realize that it is like some tubules. If you magnify further under TEM or high resolution SEM, then you will see certainly able to see the tubules like structure, and which doesn't lead to or doesn't allow these droplets to stick on the surface. So second example, I am just showing the butterfly. You might have observed that beautiful color of the butterfly wing. When if you put it under the microscopic observation, you will see the wings are like this. You further modify, magnify it, it will look like this. And further, after further magnification, you just see the wings looks like this. When the light interacts with these wings, it appears differently and color changes. So, and third example, I am just quoting, all are standard one, but anyway, just to remind you these things. So gecko, they are very smoothly climbing on the surface, vertical wall, but we cannot. Why? Because of these palm lamella. These gecko have adhesive lamella at its palm. If you magnify this lamella under microscope, then it looks like this. If you further magnify it, it looks like these tubes, which hold them on the or stick or allow this gecko to move on the vertical surface even. As we know, and from the earlier two speakers, they might have given you the idea about nanotechnology is multidisciplinary and where everyone has the opportunity to contribute something significantly, whether you belongs to physics, chemistry, or science as a whole, biology or biochemistry, even medicine, food technologist, biotechnologist, even material scientist, information technology, agriculture engineering, photonics, electronics, and many engineering branches. So everyone can contribute significantly towards nanotechnology or development. But thing is that we must know the nanoscience and technology for knowing what is happening, why they are displaying such type, why they are behaving differently than macroscopic particle of same material. For this purpose, you must have to know the science behind nano, and for knowing the science behind nano, we must have to know the science of atoms and molecules. And for knowing science behind atoms and molecules, you must know the science of electrons. So reversibly, if you know the science of electrons, you know the science of behavior or property, ultimate property of atoms and molecules. And if you know the science behind atoms and molecules, you certainly dealt with nanoscience and technology. So nanoscience and engineering can effectively improve our lives. So basically, nanotechnology is nothing but it is manipulation of matter at nanoscale to create some novel structures, devices, and systems. We have structures like material, different types of material, whether it is metallic, compound, the oxide material, or chalcogenides, or even composites and nano composites or something like that. So if you want to use such material, you can fabricate certain devices like sensors, piezoelectric sensors, pyroelectric sensors, humidity sensors, SARS sensors, whatever, any kinds of devices you can make. If you combine these devices to make automatic system as a whole, like names, nano electromechanical devices. I've just quoted one example, nano electromechanical devices. If you put some pressure, it will give you some electrical signal. So it has so nanotechnology is nothing but it is the, to my view, the practicing material science and technology at nano scale. So we have three terminology usually you mentioned in nanotechnological field. One is nanoscience. Nanoscience basically gives us or provides us the knowledge or fundamental knowledge. Many the students who are doing nanotechnology, especially physicists, chemists, or biologists, they can do nanoscience and contribute effectively towards nanotechnology. And secondly, the nano engineering, which provide you the fabrication methods, because method plays very important role. As I told you in earlier slide, that by preparing a single phase material, then only uh, you can see the ultimate effect or ultimate property of the particular material in particular devices. So nano engineering is very important in that sense. Here chemistry people, biology people, biotechnology people, even chemical engineers or mechanical engineers, all have something to do in nano engineering. And lastly, the nanotechnology, which provides us the objective basically, whether you are doing sensor technology or even many kinds of applications like medical applications or engineering applications or whatever. So everywhere nanotechnology, if you are doing that. But thing is that all the thing, we cannot bifurcate that this is nanotechnology, this is nano engineering or nanoscience. All are interrelated. So just to realize how big and how small nanomaterials are, you just see, suppose a football whose structure is 1 meter. Of course, this is very less than 1 meter, but suppose in comparison to Earth, it is 1 meter. So in comparison to our Mother Earth, it is 10 to the power 7 times smaller structure. And if you compare it with a carbon C60 molecule or say Kajal, something like that, whose diameter is around 10 to the power minus 9 meter. So in comparison to football, it is 10 to the power 10 to the power 9 times smaller structure. So you just see how small this nano structure in comparison to our Mother Earth. So it is nothing but nano is 1 billionth, a prefix to my mind. Keep this prefix in mind. So if you see the length scale, suppose we have, see, we have 3 fundamental quantities in physics. We generally teach at +2 level or 10th level, that 3 fundamental quantity: mass, length, and time. So nano is nothing but it is just prefix like milli, micro, nano, pico, or femto. If you go towards positive power side of 10, then kilometer, and so you just see the power of 10 minus 3, it is millimeter, minus 6 micrometer, minus 9 nanometer, and so on. If you go down towards or left towards this power, then so certainly I'm just showing you, convincing you that nano is nothing but it is a prefix like millimeter or micrometer or whatever. So if you allow them to put with length scale, then it will become nanometer or something. So this is just length scale which provides us or differentiate us that this is material science and technology and this is nanotechnology, nothing else. This is basically material science. We practice it at nano scale, that's all. Like we have time scale, say microsecond, millisecond, nanosecond, or something like that. We are playing with the length scale, that's all. So scale changes everything. So like human, around 1 meter or something like that. Similarly, if you go down towards this millimeter size, this ant or something like that, if you go down the size, then coronavirus, including coronavirus, also come at 10 to the power 700 nanometer or 200 nanometer, and so. Similarly, atoms and molecular size are in femtometer or angstrom or something like that. So, but thing is that we have in universe enormous scale change and differences. So at every scale, different forces dominate and different models explain those phenomena. So these things are very important. As a physicist, I am telling. So there are 4 ways. Another ways also, but I am summarizing, say, just to keep you updated. See, 4 different ways to explain different nanomaterial, why they are uh, showing different property than macro scale particle, because gravitational force become negligible at and electromagnetic force dominate at nanoscale, and quantum mechanics is the better model used to describe the motion and energy instead of classical mechanical model in nanoscale. Greater surface to volume ratios come into picture, and random molecular motion, like if you put some small particle in air, it will move randomly, and Brownian motion come into, and thermal fluctuation will come into picture. Just showing one example, just Dr. Priyanka told that shape and size gives us or provides us the ultimate effect on the nanotechnological applications. So here you can see, suppose we have taken a bead whose height and diameter are same, h is equal to d. If you elongate it, stretch it, then it will a rod like, it will take or assume a rod like structure or wire like structure. Then diameter becomes smaller and height are larger in compared to diameter. but volume are same. Similarly, if you hammer it, then it will take the or assume the shape of disc, where diameter is very large in compared to the height. Similarly, another example you just see, so surface area or shape has immense role to play in nanotechnology, mind it. And another thing, if you take a bar shaped rod, if you cut it from here, you just see surface area increases here, bottom and up. These two surfaces are extra surface we created. Similarly, you cut it for horizontally and vertically, then you have created such surfaces, and cut it so that you can increase the surface area. So I have illustrated these things in here in this example. We have, suppose we have 1 meter cube. So surface area, since it has 6 faces, we have surface area 6 meters square. If you cut it in along x direction, y direction and z direction, so we have taken together 8 such cubes and surface area increased to 12 meters square. Similarly, if you 3 cuts along x, y and z direction, then surface area increased to 18 meter. So same cube, we have surface area 6 meters square, but if you go downside, then downsizing, then we have surface area 18 meters square. So again, just reverse. You just suppose gamma is the surface area of one surface. Since we have 6 surfaces on a cube, so we have 6 gamma. To club 2 cubes together, club these 2 cubes together, it will become a bigger cube whose surfaces are now 10. So 10 gamma will be the surface energy. So this is how surface shape and size plays important role in nanotechnology. So whether it is catalytic effect or bio effect or something like that, but thing is that this is how the melting point is getting affected by nano sizing. Just one example I am showing. In the macro scale particle, you just see the materials are totally inside the surface. Large amount of material are totally inside the material. But here when we go to nano scale, the mostly all the particles come on the surface. So this plays big effect on the property. I'm just showing this why melting point goes down. You just see, if you magnify that picture, nano material, then you just take this example of this surface atom. You just see, this is surrounded by 3 atoms, and while this is surrounded by 7 atoms. So certainly, if you want to eject this atom from its surface, less energy is required. So that is how not even melting point and different energy or different properties of material changes at nano scale. So I'm just summarizing few applications. It is very difficult for a person to summarize all the applications, but I'm giving you few examples. Like in medicine and health, people have talked already. I will not go into information technology, like giant magnetoresistance hard disk. You can increase enormously the storing capacity. Similarly, energy and production sector, we have hydrogen fuel efficiency increases and consumption goes down. Material science, lightweight and strong devices we can expect from this. Food and water and environment, there are large applications exponentially growing day by day, the applications in these sectors. And instruments, you might be realizing without instrument we cannot do material science or even nanotechnology. We have to characterize, we have to implement everything. So instrument plays very important role, and even nanotechnology or nanomaterials are playing very important role in instrumentation sector or engineering also. Information technology, nanotechnology plays very important role. It will become smaller, faster, more energy efficient and powerful in computing and other IT based system. in energy sector, more efficient and cost effective technology for energy production like solar energy, fuel cell batteries, biofuel, and etc. We have in medicine already talked many things about medicine, so I am skipping. uh, Consumer goods, we have many consumer goods. L'Oreal and other uh, have already launched many products which contains nanomaterials inside, whether it is the matter of food technology and beverages, whether it is advanced packaging materials, sensors, or lab on chip or food quality testing, whatever, everywhere nano have shown their presence. So appliances and textile industry like stain proof, waterproof, and wrinkle free textiles nowadays in market. Similarly, household and cosmetic industry have also get benefited out of nanotechnology like self-cleaning, stain-free, stretch-free product, paints, and better cosmetics. Agriculture industry and agriculture sector, environmental sector, poultry industry, and many more have got benefited out of nanotechnology. And there are many exponential opportunity are still waiting to come in using nano material or nanotechnology. This is printed on a polymer, the antenna, uh, by 3D technique. Uh, this is all known. Zinc oxide nano wires are already embedded in textile industry for giving you the piezoelectric effect. Piezoelectric is nothing but if you apply pressure, then it gives you or it generates electrical potential. So your smart clothing maybe piezoelectric or nano piezoelectric rather can generate electricity uh, by walking or by exercising. You can put these clothes or put these devices on your warm palm or everywhere so that we can extract energy or generate energy from the clothing or devices. Next, the paint technology, the smart buildings. Actually, people nowadays are utilizing titanium dioxide nanoparticle in their paints. So what happens uh, when the building are being exposed by ultraviolet light? Titanium dioxide nanoparticle in paint breaks down the organic and inorganic pollutants and wash off in rain. And not only this, it decomposes the air polluting gases like formaldehyde and trap cox, SOx, NOx, and bacteria and fungi. So smart buildings are nowadays utilizing, and which are the building as a air purifier. It also works as a air purifier. And another advantage of putting this titanium dioxide nanoparticle inside paint, it it grab the property of long lasting anti-fade, and it prevents the mildew, white mildew, powdery mildew, and something like that. So, and another example of nanotechnology is in cosmetic industry. You might have seen in early days in cricket ground, people used such type of sunscreen which appears to white. But nowadays, you hardly see such type of white patches on their faces. Why? Because they use L'Oreal or Sunwax. They launched the nano scale zinc oxide based sunscreen. So if you allow this, why nano screen, why they are appearing white, basically why it is seen transparent? Because nano size zinc oxide blocks UV, and it is so small compared to the wavelength of visible light that they don't scatter it, so appears clear. That is why face appears clear. And another example, this old lady, if you use say anti-aging cream nowadays available in market, so it appears younger. So these are the examples, and some contributions from our lab also. We have made some contribution towards nanotechnology in water remediation and treatment. See, it is well known that irons are used for purification of water, underground water, groundwater, since ages. But if you use iron nanoparticle along with palladium, then it reduce or it absorbs the arsenic. So it binds arsenic, and arsenic prone area, they will uh, find better water, better quality water. So this is one of the example one can work on to it. Another is zinc oxide nanoparticle in water filtration and purification. So I am just demonstrating one experiment at MSc level. We gave one student just you just see, this is iron sulfide and sulfuric acid, so that we have H2S gas coming from this funnel to this flask, and here we have nano scale zinc oxide. So since nano scale zinc oxide has property to absorb H2S gas, so all the H2S gas coming from this absorb on the surface, and after 20 minutes this lead acetate solution turns blackening. It means when we allow H2S gas to pass on the surface of, through the surface of zinc oxide nanoparticle, it holds these H2S gas not to come in contact with lead sulfide solution, lead acetate solution, to turn black. So it can be very good water purifier where some gaseous emissions are coming, like H2S gas or something like that. It effectively nullify the bad odors also. You just see here, if you spray some colloidal zinc oxide nanoparticle, these such odors get subsides. So this has got published here in 2009. Another water purification, students can do such experiment in their lab. Carbon coated alumina. You just three parameters taken into account: pH, TSS, and TDS. You just see, we took two types of water, underground water and river water, so that you just see after filtering these water, you just see filter water, TSS, pH, and TDS, drastic difference in that. So water purification can be done, effectively done by carbon coated alumina. Another example is that you might be seeing 4 bottles. These are nothing but these are different color ink: red, blue, green, and black. So we functionalize these ink with our own nano materials. So what happened? The effect of which, it gives you shiny. Another thing is that it will stick on the surface for longer time, and it will not fade, as well as bacterial and fungal effect will be nullified. We have also worked, our students have worked towards the 3 and 4 ply face mask. Just see the nanomaterial already talked, so I'm not going into the detail with this. This is certainly very boon for doctors who is doing autopsy, clinician, municipal worker, and cox, knox, bikers, and etc. So I'm not stressing upon this slide because already talked. Another thing, for gauze and bandages, we keeping in mind those things, we antibacterial cotton cloth fabric. You just see the different types of nanomaterial sticked on the surface by coating. So, and effect of which you can see. So it effectively nullify the Staphylococcus aureus. So certainly it will be a boon for cotton industry and dressing, and certainly like that. And some skin care products have also been developed. I'm not taking uttering name because some patent has been filed on such type of thing. That is why I'm not uttering the name and how we have prepared. But thing is that they can. So even herpes or wound can be effectively treated by nano materials. This is one of the example in our lab. This is acid burn case. Acid dropped on its the student's finger. You just see after just applying our nano based product, colloidal nano based product, functionalized of course. You just see after 20 minutes the same finger, you just see the effect. Of course, all of you are well familiar that it has very anti-cancerous effect. So we have published this paper in this. Those who are interested in going through the details, you can go through this paper. Another is the energy harvesting. See, what we did here is, you just see here test material. Test material is nothing but it is nano composite. This is fabricated in our lab, and this is DSO. This DSO is nothing but digital storage oscilloscope. If you apply this weight onto or hammer it, then certainly, since it is piezoelectric material, so it will develop some charge, and this charge will be taken care of this digital storage oscilloscope. So charge signal will be seen here, and potential will be, or voltage will be developed. So you just see, we use NBT, sodium bismuth titanate, and PVDF nano composite. It has just appeared in Journal of Material Science, Materials in Electronics this year. You just see this voltage increases if you increase the concentration of NBT here. So signal strength increases with composition and shift towards right side with the NBT content. So if you increase the force or impact energy, then voltage increase, and if you increase the content, then also voltage increases. So signal strength or contact voltage increases with height of impact as well as NBT content. Similarly, the figure of merit is very important parameter. How much mechanical energy we have applied and how much energy we are receiving in terms of electrical energy, that is very important. So because we have to use it in sensor or energy producing devices. So keeping these things in mind, we could develop this NBT based material composite. So generated energy increases with height of impact. You just see energy increases, and with NBT content also, you just see here energy gets increased after increasing the NBT content. This is why they are giving you voltage. Since such materials are perovskite one, so it contains dipoles. When impacted, all dipoles will tend to align themselves in this direction. So polarize goes up and give you voltage up signal, and by releasing them, then again it will come back these dipoles and give you the negative signal. So this is how voltage increases if you load it or impact it. So So present such type of composite used in structure building, structure health monitoring. These structure is nothing but skyscraper buildings, multi-story building, bridges. So these health monitoring of those things, hanging structures, and different sensing and energy generation applications. See, you might have realized that voltage generation was not regular. So just to make it regular, we use filter technology, what we use in electrical engineering or electronics. So we use capacitor and RC filter. So you just see the voltage generated through this technique is quite constant. So that is why this can effectively be used for energy generation. And if you increase the capacitor value of C, the filter, this C, then this value goes on decreasing. Blue color you just see, capacitance increases, this voltage decreases. Similarly, you can, I'm just, this slide is especially for students. You can make such type of sensors very easily in your own laboratory. I555 triple timer circuit. This is very cheap IC. Only 50 rupees, costing 50 rupees. You can connect it like this, and you just apply your own product like nano composite or ceramic material, nano ceramic here, and drop something, or you apply some pressure. It will sense here the width of this pulse at voltage output, this CRO will be seen. So by putting here pressure, increasing the voltage. So this way you can easily make sensors out of it. So applying pressure, you put these things here, pin number 6 or 1, so you can make sensors. So this is only for students. This has also, so those who are interested in studying such type of thing or doing such type of thing, you can go through this, or you can contact me at my scale. So my aim is to let students aware that nano fabrication method or method of fabrication is more important. Then only you can apply it for applications. So we have basically two types of nano fabrication method. I am quickly showing these things because I know that time is passing. So I will just show you slides. I will not go into detail about the X-ray or TEM or whatever. I'm showing that how materials are being fabricated. So we have two approaches, top down and bottom up. Top down approach, bigger size particle goes into a smaller size particle. Bottom up approach, say supramolecular chemistry is the best example. Say smaller particles combine together, so become larger particle. This can be done by crushing the particle. This can be the fullerene molecules, the kajal, what we use in our eye. So certainly shape, as I shown you earlier, that shape and size gives you different effect. You just see the UV visible spectroscopy result. You just see AU spherical nanoparticle gives you lower wavelength excitation, while rod shape give you the higher side, show you the higher side excitation. But thing is that being a student or being beginner, you must take lesson from earlier technology that I wanted to show from this slide, that past failure to consider environmental consequences. So only developing technology is not important. Importance is also, stress should also be given on its environmental consequences, because we have already already maybe 50 or 55 or something like that, but coming generation must not forget you if you done or committed some mistakes. So environmental consequences has large impact So like we have semiconductor industry, metal solvent and waste, synthetic chemicals like DDT or something, application of natural like chlorine or asbestos. Similarly, transportation energy, of course, energy sector is very important energy, but at the same time, we must see the air pollution, global warming, nuclear waste, and many more things to discuss. See, we Indians especially minded, small adjustment in early in the trajectory of technology have large consequences. Repeated technology like just a small adjustment in the technology has large consequence. So we must introduce at government level, at NGO level, or at any level, university level, the environmental perspective early into the culture of engineering, emerging technology, like in innovation, research, and education. It should be incorporated in the innovation, research, and educational mission. You see the impact of e-waste and e-trace. I will not see, these are some cartoons since time is less. See, earlier the trends of material preparation in 19th century, early chemistry, toxic ingredients were used. In 20th century, we call it brown chemistry, where lower toxic pollutants are there, or waste reductions. We work on waste reduction, but in 21st century, we are going towards green chemistry and biotechnological approach, combining with nanotechnology and new materials and renewable. We use renewable sources, aqueous solvent, clean manufacturing process, and eliminate the toxicity, of course. So uh, biosynthesis or biotechnological approach, we call it green chemistry. There are various environmental benefits, safe and controllable, water, aqueous things, waste stream reduction, biorenewable, and reaction selectivity opportunity. So we have basically three approaches for nanosynthesis. One is physical method. Under physical method, we have various things like ball milling technique, thermal evaporation, lithographic technique, vapor phase epitaxial growth, strain layer growth, and so on and so forth. Then chemical technique, we have various chemical technique like sol gel, micellar medium, hydrothermal emulsion precipitation, solution based chemistry like water, oil water, air water interface, oil interface, and polar non-polar organic interface and solvent, so that we can produce using chemical technology various nanomaterials. Then comes the biological methods. In biological methods, we generally use bacteria, fungi, yeast, plant extract, fruit juice, food beneficiaries. We call it as nature's nanofactory because it is gifted item from the nature. So we call it this. So my question is, can animal, see, when yeast and microbes, plant system and other things are contributing towards nanotechnology, can animal or their parts too can participate in nanotechnology? That I will answer yes. It all the games of energy. See, whatever be the technique you apply for nanomaterials development or production, it all the game of energy. Keep in mind the game of energy. You have to produce some kind of energy so that they can we can produce various variety of nanomaterials either using top down or bottom up approach. Details you can see from our book recently published from Springer in 2018. You can go through it. One thing which very soon will be introduced in the market is 3D printing technology, which generate nano devices. So nanotechnology, this is printable desktop. This is of course imaginary one. So portable desktop size nano factory. This is just artist, one can say, imagination. But it will come into market very soon. You can use nanotechnology not just for producing new product, rather a new means of production. Manufacturing system, this is a manufacturing system, makes more manufacturing system. See, system alone can manufacture different systems. So these things you must keep in mind and work in this area also. This is open area nowadays, 3D printing. So we are, so being biotechnology, or most of the audience are belonging to this biotechnology and engineering side. So nanobiotechnology area is nowadays upcoming field, considered to be upcoming field. This is nothing but it is the connection between nanomaterials and biologicals. So the contributing field, anyone can contribute, as we discussed earlier, physics, chemistry, or material science, or biology, biotechnologies, and anyone. So question arises, nanobiotechnology, can we have device fabrication? Of course, as Dr. Priyanka told, that affinity chromatography, if you suffer or if you allow such material through affinity chromatography, certainly cell debris will be bifurcated and we have pure material in our hand, and we can produce many consumer beneficiaries products you can think of. So this slide I am skipping because this is standard, because time is less. So one thing must keep in mind being biologist, so length scale, because length scale which ultimately decides the fate of the produced nanomaterials, but this is nanoengineering. So these things you can always use for producing or for killing, for producing the nanomaterial or for killing such virus or cells. So you must know the length scale. So biosynthesis is not very complicated thing. You just select the biological system first and then prepare the culture medium and regulate the medium condition by adjusting pH and other things, temperature, and you just inoculate your biomaterial material you want to produce, like silver, gold, or whatever, and filter it so that you have material in your hand. Of course, this is not the proper time to discuss thermodynamics behind it and all the things, but you may read, interested reader may read these things in our publications. But only thing I wanted to show you that, especially right hand side, how free energy is generated inside the system, why microbes are able to produce nanomaterial, why plant systems are producing nanomaterial, why animal systems are contributing significantly towards nanotechnology. Or answers are these things. See, there are various chemical and biochemical resonance energy, tautomerization, glycolysis, free energy from organic acid, generation of ROS, SOD response, oxidase and reductase, protein chemistry, and many enzyme reactions and other biochemical process basically generate energy inside the system so that we have different nanomaterials in our hand. We have to regulate the medium condition by temperature, pH, precursor concentration, and other things. This is also in our book. This is easily available on Google. You just make it. So we could produce material, different kinds of material, using microbes, plant parts, flower, fruit or fruit peels, food beneficiary like ghee and honey, uh, animal discards, expiry medicines and drugs, soft chemicals, etc. And materials type, if you discuss about materials type, then we could produce colloidals, metals, oxides, chalcogenides, and composites using this. These are the list what we have produced using such technique. We have collaboration with these things, JNU, IIT Mumbai, NPL, HEG Bhopal, and S Hospital Pune, IGIMS Patna, and other also. So I'm just, from now, I am just showing the slides. I will not discuss more. I'm just showing how microbes could produce the material, titanium nanoparticle from microbes, lactobacilli. This is cadmium nanoparticle. All are published. You can go through them. This barium titanate nanoparticle in colloids and surfaces 2010 paper in from microbes, yeast of course. And this is the biochemistry basically we did, how energy are generated. But I will not discuss this into detail because we have no sufficient time. Then microbes, yeast assisted synthesis. We just see titanium dioxide nanoparticle. Are you listening me?
Yes, sir. Now you are audible.
Okay. So barium titanate nanoparticle from yeast Saccharomyces cerevisiae. These have published in integrated ferroelectric colloids and surfaces and so on. So then chalcogenides, cadmium sulfide nanoparticle, J colloids and interface science through yeast and lactobacilli. This is magnetic nanoparticle. Again, why they are producing? This is the chemistry. This is already discussed in our book. Who readers who are interested, you can go through them. Then plant assisted synthesis. See, plant has beauty, or plant part has beauty, that it contains not only the phytochemicals, rather it has various types of phytochemicals present in different plants or plant parts, like polyphenols, flavonoids and terpenoids, functional group for each type, which is being which are used as stabilizing agent. Then we have polyphenols reducing sugar, functional groups, proteins and other things which are being taken use as reducing agent. And then some chemicals present in biochemicals rather present in the plant system is organic acids like citric acid or functional groups, enzymes, etc., which are acting as the capping agent. So plant system are considered to be the better system for producing different variety of nanomaterials. There are a plethora of plant system and plethora of nanomaterials could be produced using this. Only thing is that you have to have water soluble salt used for this purpose like this. Then plant assisted synthesis, we took three kinds of plants: xerophytes, mesophytes and hydrophytes, and results are that here. We could produce nanomaterials out of bhringraj, silver nanoparticle from bhringraj. Similarly, this is cycas. This is belpatra. Of course, belpatra contains various kinds of coumarins, terpenoids and organic acids, which are responsible for converting gold, macroscopic gold into nano gold. Similarly, gold nanoparticle from three types of leaf: mentha, eucalyptus and ocimum, tulsi. These are the chemistry or energy generation behind these three types of plants. Cycas. Then comes fruit assisted. See, from this flower, rose, we could produce gold nanoparticle for different aim, for cosmetic industry or something like that. This is orange peel, which we could produce silver and zinc oxide nanoparticle, and this is the chemistry. Similarly, this is chili, green chili, which could produce the silver nanoparticle, or a platinum nanoparticle from pomegranate. You just see very beautiful X-ray and this. And come to the drug assisted nanoparticle. See, we could we procure lactobacilli tablet, sporogen tablet, which is probiotic from the market, and same result or similar result we could produce what microbes lactobacilli could produce for us. This is titanium dioxide nanoparticle, and zinc oxide nanoparticle. Then what is new about it? See, what we did is we collected scrap material from the market, iron scrap, and we collected the expiry medicines. So expiry medicines we could produce, you just see, very good iron nanoparticle and zinc oxide nanoparticle. So from scrap. So this is based out of waste, you can say.
Sorry, sir. Sorry to interrupt you, sir.
Right.
Just wanted to understand how many more slides do we have, because we have another webinar at 5. So just
five slides. Just
okay, sir.
five.
Okay, sir.
Okay.
No problem, sir. Okay.
Okay. So I'm just skipping this. Okay. Okay. So this slide basically I wanted to show you all. Can animal also contribute in nanotechnology? Animal or animal discard or part. You just see here the arthropod, the cockroach. So we have not we have collected this cockroach from our kitchen, dead cockroach, and we could produce such type of material. Many similarly, banana fly could produce nano material for us, and this is household mosquito could produce cadmium sulfide nanoparticle for us, and surprisingly we could use this for technological development. I see time is short, I can show, but anyway I am skipping those things. So this is fish discard, and silver nanoparticle could be fabricated using this. The goat slaughterhouse waste, this, and we could produce nanoparticle, different nanoparticle. Chicken slaughterhouse, chicken processing discard, we could produce material. So animal can effectively say, this is the last slide. So nanotechnology, basically I want to discuss some scope. See, manpower development. Nanotechnology is not only to develop different devices or different material. Rather, our aim should be to develop manpower also, who could train the youngers. So manpower development also is an aim of nanotechnology. In higher education, research and development, educationists and trainers, entrepreneurship development programs, and youth training. So this should be a mission. So at government level, at university level, at research R&D level, and so on. So it ensures clean and green environment, waste recycling, and so on and so forth. Medical and health sector, better lifestyle. Agriculture and food sector will especially benefited out of nanotechnology. And of course, the employment generation and new opportunities are still waiting the nanotechnology field, and economical and sustainable growth, small and medium scale industry could be done. So certainly if we combine industry development and manpower development, so training will come into picture. Manpower development and product development, then education should come into picture, and R&D. See, industrial development and product development, R&D should come into. So taken together, we can, if you have 3 wings, then we can grow as a nanotechnology as a whole. So nanotechnology offers great promises for the future. So I will fail in my duty if I will not utter few words about our group. This is my friend Anal Kant Jha. This is Professor A.R. Kulkarni in IIT Mumbai. Amit Kumar, who is a professor at BIT Patna. And these are the group of doctors who is contributing towards for validating our nano compositions. So that, and thank you. So this is all about my talk. So
Thank you, sir.
okay.
Thank you, Professor Kamalkar Prasad, sir, for your enlightening talk. I'm sorry, we do not have time for questions, but you can definitely post them to
Okay,
me on
okay. Thank you.
on the WhatsApp group, or you can mail it to me,
Okay, hi, yes, yes.
and we will get it
Thank you.
answered by sir.
Okay.
Uh, with this, we will just wrap up with a small vote of thanks. Elizabeth, please go ahead.
Okay, ma'am. Thank you. On behalf of Department of Medical Biotechnology, I extend a very hearty vote of thanks to all our eminent speakers for sparing time from their busy schedule and gracing us with your talk and sharing with us your knowledge on nanotechnology and its various future prospects. Thank you for making such excellent presentation and making the seminar interesting and meaningful. I'm immensely thankful for our director and HOD, Dr. Mansi Thakur, for always being there and supporting and encouraging us for organizing such a meaningful webinar. Finally, the wonderful participants who have joined us in this webinar, not only from our department, but from also other colleges. Thank you all for your cooperation. I'm sure that all the participants who have attended today's webinar would use all of this gained knowledge into finding their way into the field of nanotechnology. Last but not the least, I would like to thank the entire SBS team and Mr. Anil for their IT support as well as encouraging us. And I request a few minutes from all of you to send your feedback on our event. The link has been sent to your WhatsApp as well as your email ID. And the e-certificates to all the participants who attended to today's webinar will be sent via email by next week. Thank you everyone for joining us. Thank you.
Thank you.
Thank you, everyone. Thank you, sir.
Thank you.
Thank
Okay.
Thank you, sir.